Welcome to video 3 in the series on insect herbivores. In this video, we're going to explore some of the chemical defenses that plants have evolved to ward off hungry would-be herbivores, and some of the countermeasures that insects have evolved to get around them. These chemical compounds, which we call secondary metabolites, serve as a key line of biochemical defense against the onslaught of herbivory. Okay, first, a metabolite is just a name for a molecule or a compound that's used in metabolic processes. A primary metabolite is a molecule or compound that's essential for plant growth and reproduction. A secondary metabolite is a molecule or compound that isn't directly involved in the plant's basic metabolic processes. Instead, these secondary metabolites are involved in things like defense, pollinator attraction, or other ecological interactions. In terms of plant defense, secondary metabolites can disrupt herbivore physiological functions and inhibit their feeding, or even just repel them altogether. Here, we'll take a look at a few different categories of these defensive compounds and explore how and why they're so effective. Plant chemical defenses can be divided into two broad categories: qualitative defenses and quantitative defenses. Qualitative defenses work by virtue of their high-impact chemical properties that make them especially toxic or poisonous to insects. Quantitative defenses, on the other hand, rely on the sheer quantity of a given compound, reducing the palatability or the digestibility of plant tissues through high concentrations. Let me give you some examples of each. Nicotine is a pyridine alkaloid found in tobacco plants. This compound targets the nervous system by mimicking a neurotransmitter called acetylcholine, and it overstimulates the neural synapses and then leads to convulsion, paralysis, and ultimately death. Nicotine's rapid action mirrors that of modern synthetic insecticides. This just underscores how plants have long been nature's chemists when it comes to the battle against herbivores. Cyanogenic compounds are another example. They're found in plants like cassava and clover. When plant tissue is damaged, these compounds release hydrogen cyanide, which disrupts the cellular respiration by blocking the cytochrome system, effectively cutting off the cell's ability to use oxygen. This, of course, results in rapid death for any insect herbivores attempting to feed on these plants. Milkweeds produce cardinalides, which are compounds that disrupt the sodium-potassium ion balance in insect cells by inhibiting the sodium-potassium ATPase enzyme. This mechanism not only deters herbivores, but can also serve as a defensive tool for specialist herbivores like the monarch butterfly caterpillars, which can sequester these compounds to make themselves more toxic to predators. Similarly, glucosinolate compounds in mustard plants degrade into toxic byproducts when the leaves are damaged, effectively targeting herbivore protein and respiratory systems. These examples highlight how qualitative defenses rely on their high toxicity and immediate effects to deter herbivory. And there are a wide variety of other qualitative defenses that have varying degrees and mechanisms of impact. Now let's consider quantitative defenses, which focus on high volume and durability rather than on rapid impact. These defenses are typically large molecules like tannins, lignins, cellulose, and silica that are present in high concentrations in plant tissue. Their primary role is to reduce the palatability and digestibility of plant material, and can often target a broad range of herbivores. Tannins, for example, are a group of polyphenolic compounds. When herbivores bite into tannin-rich tissue, these compounds form complexes with proteins and digestive enzymes, reducing the herbivore's ability to process the plant matter. While effective against many herbivores, some insects have evolved gut physiologies that neutralize the effect of tannins, showing how this evolutionary arms race between plants and herbivores never truly gets resolved. Lignin is another quantitative defense. It's a structural polymer that adds rigidity to plant cell walls. This toughness makes tissues difficult to chew and digest. Herbivores feeding on lignin-rich plants often experience high mandibular wear and slower nutrient assimilation. Latexes and resins represent yet another fascinating group of quantitative defenses. Milkweeds and spurges produce latex, which is a sticky emulsion that gums up the herbivore's mouthparts, making it physically difficult for them to feed. Resins, commonly found in conifers, serve a similar function, trapping herbivores with their stickiness, while also delivering a chemical punch through toxic terpenoids. Some of these examples of chemical defenses can be looked at from a mechanical defense perspective as well as a chemical defense perspective, since they're directly related to how physically difficult it is to consume plant matter. Quantitative defenses typically don't act swiftly like their qualitative defense counterparts do, but their cumulative effects can be just as devastating to herbivores. By making plant tissue tough to process or indigestible, these defenses reduce herbivore efficiency and growth rates, ultimately shaping herbivore populations and community dynamics. This brings us to a second way that plant defenses can be categorized. They can be constitutive or inducible. Constitutive defenses are always present, serving as a continuous barrier against herbivores. These defenses include structural features like toughened cell walls, waxy surfaces, or trichomes, as well as chemical defenses like tannins and alkaloids. Constitutive defenses ensure that the plant is always prepared for herbivore attacks. However, this constant vigilance comes at an energetic cost, as maintaining high levels of defense requires significant resources. Inducible defenses, in contrast, are triggered only when a plant detects a threat, allowing it to conserve resources during times of low herbivore pressure. A critical component of some of these inducible defenses is the hormone jasmonic acid. When a herbivore begins feeding, a small peptide hormone called cysteamine moves through the plant, signaling for the production of jasmonate molecules like jasmonic acid. This hormone activates a cascade of biochemical changes, resulting in the production of defensive compounds like proteinase inhibitors and polyphenol oxidases. These compounds disrupt herbivore digestion, reducing their ability to process plant material, effectively and efficiently deterring further feeding. The biochemical processes and defensive compounds that are initiated when jasmonic acid is released can sometimes even affect plants that aren't under attack. Research has shown that plants can detect certain volatile organic compounds released by neighboring plants that are under herbivore attack. The neighbors that aren't under attack can then preemptively activate their defensive pathways before they themselves get targeted. In agriculture, this understanding of jasmonic acid signaling has led to practical applications. For example, when crops are treated with jasmonic acid, it can trigger the plant's inducible defensive responses to help repel pests and minimize damage. This strategy has been particularly effective in plants like tomatoes, where jasmonic acid treatments have been shown to increase the levels of defensive metabolites and significantly lower herbivore damage. Plants' use of chemical defenses illustrates the complexity and adaptability of their responses to herbivory. Each compound, whether an alkaloid or a tannin or a phenolic compound or jasmonic acid, plays a significant role in deterring herbivores, whether disrupting their physiology or even recruiting allies to the plant's cause. Through these diverse biochemical strategies, plants have developed a sophisticated toolkit to manage herbivory and ensure their survival in a world filled with hungry herbivores. In subsequent videos, we'll continue to explore how plants and herbivores interact, focusing on the counter-adaptations that herbivores have evolved in response to these plant defenses, and the unavoidable evolutionary arms race that shapes not only individual species, but also entire ecosystems. Well, that's all for now. Thanks for tuning in. I'll see you next time.